Okay, now coming on to sucrose. Sucrose is a disaccharide. I just want to make you understand how it actually forms the bond. So disaccharide glucose is made up of disaccharide sucrose is made up of glucose and fructose. Here the glucose, you can see the diagram of glucose here, the structure. Oxygen present, first carbon, second carbon, third, fourth, fifth and sixth carbon here. Similarly, if you see this fructose, first carbon is this one, second, third, fourth, fifth and sixth carbon. So there is a bond formation between the first carbon of the glucose and the 1, 2, 3, 4, 5, fifth carbon of the fructose. So it is 1, 5 glycosidic bond. The bond which is formed between two sugar molecules, between two monosaccharides to give rise to a disaccharide and further is called as a glycosidic bond. So this is how we number it. Why we call it 1 5? Because the first carbon in glucose is attached to the fifth carbon in fructose. So it is 1 5 glycosidic linkage. Clear? Clear? I think this will be clear to everyone. It is very easy. Now let us discuss about the glycosidic bond in detail. Just see how it is being formed. This is, what is this? I told you this is glucose, first carbon, second, third, fourth, fifth and sixth carbon. This is fructose. Fructose here, it has been revolved. See, can you see it has been revolved? Revolved in the sense that this carbon has been given 1. So same thing, because ultimately second carbon is attached to oxygen, so we can number it this way also. Both the types of numbering is same. Here also we have made this type of diagram. Now here what happens when they are combining? When they are combining, here it has rotated in a fashion. So here what happens, two molecules come together, and this is glucose and this is fructose. When they come together, the OH and OH are attached to both the ends. Now this OH and H are removed in the form of water, so they lose a water molecule to form a bond between them, and this is called a glycosidic bond. Glycosidic bond is a bond which is formed by the removal of a water molecule and mix and joins to form a disaccharide, and further more linking is there in a polysaccharide. Clear? Now, if we talk about maltose, maltose is formed by two glucose molecules. So there also two glucose molecules will combine, one glucose plus one glucose. Similarly, in sucrose, they will be combining in such a manner. I have told you this sucrose only. So this you need to understand. You have already drawn the diagram also. Now, coming on to a complex um, this thing, polysaccharide, that is glycogen. Glycogen, I told you, is a homopolysaccharide which is having only single type of repeating units. Clear? See here the structure. What is that single kind of repeating unit? It is actually the okay, first tell me, what is glycogen? Have you heard about glycogen earlier? Yes, glycogen is stored in liver and it is the form in which glucose is stored. So now can you tell me what will be the repeating unit in glycogen? Yes, glycogen will have glucose as the repeating unit. So it is, when it is made up of only glucose, 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 glucose all linked together, it is a homopolysaccharide because it has only one type of uh, micromolecule, that is one type of unit present, that is glucose. So it is a homopolysaccharide and it also acts as a reserve food for animals, or we can call it as animal starch. If you remember, animal starch was the name which was taken by our teacher in class 9th. Especially the food is stored in case of animals and fungi in the form of glycogen. Where the glycogen is stored? In liver and in muscles. It gives you about 7% of the wet weight of liver is because of this glycogen which is present. Now there are two glucose molecules are linked together by alpha 1,4 linkage. 1,4, why we call it as 1,4? First, second, third and fourth carbon is there. Can you see here? So it is alpha 1,4 linkage. Two types of linkage can be seen. This is alpha 1,6 linkage and it is alpha 1,4 linkage. If you count it, how it happens? One first carbon is remain this one, first, second, third, fourth, fifth and sixth carbon. This is it. Now here what happens? The numbering is being done in such a manner that it gives a 4 number to this. So 1, 2, 3 and 4, fourth carbon. So this is generally formed here. But what happens when these are the single chains? When these single chains have to make a double chains or make layers upon it, then they cannot form horizontally like this. Vertically they cannot form 1,4 linkage. So in that case they will form alpha 1,6 linkage, because the first carbon here, this is considered to be the first carbon here one. So the first carbon attaches to the sixth carbon here and form a chain or layers. Clear? Are you understanding? Just look at the diagram once again. The straight long chains are formed by alpha 1,4 linkage. I'll tell you what is beta also. That is also, there is also a beta thing present that I will discuss later. Today you understand what is alpha 1,4. Alpha means straight. This kind of structure is there. Straight, the front side is being attached. I'll compare it with beta and then it will be more clear to you. For now, just understand the numbering. This was the first carbon, second, third, fourth, fifth and sixth carbon. So the first carbon is attached. First carbon of the first glucose group is attached to the fourth carbon of the second glucose group. So they are forming an alpha 1,4 linkage. So this is the long chain, single rowed long chain, all having alpha 1,4 linkage. But if it is a polysaccharide, it will not be made up of only one layer. It will probably have more layers in the top and more layers at the bottom also. So how these layers are attached? We have all the long chains and then these chains are joined together by alpha 1,6 linkage. Now see how this happens. This is alpha, this is one first carbon. So the first carbon here attaches to the sixth carbon here and forms an alpha 1,6 linkage between the two single layers of glycogen. And this is how the complexity occurs. The more complex structure is formed from these kind of structures, from these kind of linkages in all the directions. And more glycosidic bonds, more amount of glycosidic bond, more strong is the structure, more stable is the compound, and more number of bonds are present. So you understood, alpha 1,4 give rise to long straight chains and alpha 1,6 give rise to layering. That is, one chain will combine to the another chain, will be joined with the another chain by using alpha 1,6 linkage in the glycogen. I think this much conceptual thing is enough for today. So we have studied about what is biomolecule, how many types of biomolecules are there, and we have started with carbohydrates. I have told you about monosaccharides, uh, disaccharides, oligosaccharides and polysaccharides, homopolysaccharides and heteropolysaccharides. You will read on today to make the conceptual knowledge better. Now I have already discussed about glucose and fructose. Told you that glucose is made up of glucose is having aldehyde group and fructose is having ketone group. The difference between them is very important. You have written a difference table in your notebook. You need to learn that table. Then we have discussed about the glycosidic linkage, which is formed by removal of a water molecule. And then one polysaccharide we have discussed today, that is glycogen. You will write down these notes or paste the printouts as per your wish and start learning the notes simultaneously, and will ask in case of any confusion in the next class. Thank you so much.